Columbus did not discover America. And the proof is not a legend, not a saga, not a broken shield buried in the dirt. It's in the DNA of a woman who died nearly 1,000 years ago. Her genetic code does not belong in Europe. It does not fit medieval Scandinavia. It does not align with anything historians expected to find in Viking graves. And yet it is there, embedded in the bloodlines of Iceland. If this evidence holds, and it increasingly does, then the timeline taught in classrooms across the world is not just incomplete. It is wrong. For centuries, the story seemed stable. In 1492, Columbus sailed west and opened the gates of the New World to Europe. Whatever complexities followed, colonization, catastrophe, empires, they all began there. That date became a fixed point in global memory. But history has a weakness. It relies on records. And records are written by survivors, by winners, by those who choose what deserves preservation. DNA does not choose. It simply persists. In the last 2 decades, genetic science has evolved from a supporting tool into a forensic time machine. Fragments of bone, preserved teeth, and even soil can now yield molecular sequences that reveal ancestry with startling precision. Entire migrations are reconstructed from patterns invisible to the naked eye. When researchers began applying these techniques to Viking Age remains, they expected confirmation of known Scandinavian movements. Norway to Iceland, Iceland to Greenland, Greenland to the edges of North America. That much had already been suggested by archaeology and saga literature. But what they found went further. In Iceland, a country with one of the most thoroughly documented genealogical records in the world, scientists identified a mitochondrial DNA lineage that did not originate in Europe. It did not trace to the British Isles, where Vikings often took captives. It did not match any known medieval Scandinavian population. Instead, it matched indigenous populations of North America. The date of its introduction? Around the year 1000, 5 centuries before Columbus. Let that sink in. This was not a rumor carved into parchment. This was not a poetic exaggeration from a saga. This was biological evidence that a Native American woman lived in Viking society, and that her descendants survived for generations. Somewhere, sometime, across the North Atlantic, a human exchange occurred, and that exchange left a molecular signature. To grasp how destabilizing this is, we need to revisit what we thought we knew. Yes, archaeologists confirmed the Norse presence at Meadows in Newfoundland decades ago. Timber structures, iron nails, evidence of ship repair, radiocarbon dating, and tree-ring analysis placing the site around 1021 CE. But the dominant interpretation was cautious. A temporary camp, a brief venture, a failed colony that vanished without consequence. An experiment, not a transformation. DNA is suggesting otherwise, because if a woman of indigenous American ancestry ended up in Iceland around that same period, it means the voyage was not just a landing. It was a return. Vikings did not simply arrive and retreat. They interacted. They transported people. They crossed the ocean in both directions. That changes everything. Imagine the scale of that journey. A wooden ship, open to Atlantic storms. Weeks at sea. Navigation guided by stars, currents, and instinct. No modern maps. No certainty of survival. And yet they did it. Not once, but enough times to establish settlement attempts in Greenland and North America. The sagas speak of lands called Helluland, Markland, and Vinland. For centuries, scholars debated whether these were metaphors or myths. Then archaeology confirmed their plausibility. Now genetics is confirming their consequence. The Vikings were not accidental tourists. They were capable navigators, pushing into unknown waters with intention. But here is where the story grows more complex. When the genetic anomaly was first identified in Iceland, skepticism followed immediately. Critics suggested later migration, clerical errors in genealogical records, modern contamination of samples, statistical flukes. Science thrives on doubt, so researchers tested again. Independent labs reanalyzed sequences. Comparative databases expanded. Authentication protocols strengthened. And the signal remained. The mitochondrial lineage was ancient. Its introduction aligned with the Viking Age. Its closest genetic relatives, were indigenous populations of northeastern North America. The data refuse to disappear, which forces us to confront an uncomfortable question. If Vikings reached America first, not symbolically, not speculatively, but biologically, why did their story not define global history? The answer may lie in scale. Columbus arrived backed by monarchies, armed with imperial ambition, followed by fleets and colonization machinery. His voyages triggered sustained European expansion, reshaping continents. Viking expeditions were different. They were decentralized, personal, often experimental, driven by land hunger, trade, exile. They did not return to Europe with proclamations of empire. They returned with stories, and perhaps with people. That subtlety may be why their impact faded from dominant narratives. But subtle does not mean insignificant. Consider what this means for indigenous history as well. Contact between Europe and the Americas may have occurred without the immediate cascade of colonization. There may have been moments of exchange that did not erupt into conquest. Trade, conflict, curiosity, movement, human encounters that were neither pure myth nor full-scale invasion. The DNA suggests complexity where textbooks prefer simplicity, and the implications ripple outward. If Vikings transported individuals across the Atlantic around 1000 CE, then transatlantic navigation was not an accident achieved by Renaissance technology. It was a capability achieved centuries earlier by seafaring societies operating with far fewer resources. The Atlantic was not a barrier awaiting breakthrough. It was a corridor navigated by those daring enough to attempt it. As researchers continue sequencing remains from Norse settlements in Greenland and exploring new techniques for extracting DNA from ancient soil layers in Newfoundland, more fragments of this puzzle may emerge. Every sample carries potential revelation. Every strand of preserved genetic code challenges a narrative carved in stone. This is not about diminishing Columbus or rewriting history for shock value. It is about evidence. Because when molecular data contradicts established timelines, historians are forced to reconsider foundations. And right now, the foundation is trembling. In the next part of this investigation, we will examine exactly how scientists extract ancient DNA from thousand-year-old remains, how they authenticate it, and how they differentiate genuine Viking-age genetic signatures from later contamination. We will follow the methodology step by step, because extraordinary claims require extraordinary evidence. But if the evidence continues to accumulate, if the genetic traces from Iceland, Greenland, and North America align, then we are looking at more than an interesting footnote. We are looking at proof that 5 centuries before Columbus crossed the Atlantic, Vikings had already bridged it, not in legend, in blood. And once blood enters the historical record, it is very difficult to erase. To understand why this discovery matters, we have to move from the headline to the laboratory. Because ancient DNA is not a romantic idea. It is not a dramatic revelation pulled from a single bone under a spotlight. It is a discipline built on contamination control, statistical modeling, replication, and doubt. When scientists claim that a 1000-year-old mitochondrial lineage in Iceland traces back to Indigenous North America, they are not relying on intuition. They are relying on a chain of procedures designed specifically to eliminate error. The process begins with extraction. Ancient DNA does not survive easily. Heat, moisture, bacteria, and time break it into fragments so small they can resemble background noise. What remains is often damaged, chemically altered at the ends, riddled with breaks. The average fragment length can be shorter than 100 base pairs. That damage, ironically, becomes part of the authentication, because modern contamination looks different. When researchers work with ancient remains, bone, tooth pulp, sometimes even preserved hair, they do so in clean rooms with positive air pressure, protective suits, UV sterilization, dedicated equipment never exposed to modern DNA samples. A single stray skin cell from a technician could compromise results. So protocols are strict. The outer layer of bone is removed mechanically to eliminate surface contamination. The interior is powdered. Chemical buffers release whatever DNA fragments remain trapped in the mineral matrix. Those fragments are then isolated and prepared for sequencing. What emerges is not a complete genome. It is a collection of broken pieces. Bioinformatic algorithms assemble these fragments, comparing them to reference databases of known human populations. Patterns begin to form. Mitochondrial DNA is often the first focus because it exists in multiple copies per cell, increasing the chance of recovery. Unlike nuclear DNA, which mixes each generation, mitochondrial DNA is passed almost exclusively from mother to child, forming stable maternal lineages across centuries. That stability is what allowed researchers in Iceland to identify something unexpected. In a detailed study of Icelandic mitochondrial lineages, scientists noticed one cluster that did not align with European haplogroups common in medieval Scandinavia. Instead, its closest relatives were found among indigenous populations of northeastern North America. At first glance, it could have been dismissed as a later introduction, but genealogical tracing placed its arrival around the Viking Age. That dating was not guesswork. Iceland's historical records are unusually complete. Family lineages have been preserved in written sources and church documents for centuries. When combined with genetic analysis, researchers could trace the lineage backward generation by generation until it converged in the early 11th century. This was the critical point. If the lineage had entered Iceland in the 17th or 18th century through later contact, records would reflect it. If it were a modern contamination artifact, the molecular damage patterns would not match ancient DNA signatures. But the molecular damage was consistent with age, and the genealogical convergence aligned with Viking expansion. Still, extraordinary claims demand resistance. Some critics propose that the lineage might have originated in Europe, but simply resembled Native American haplogroups due to limited database sampling. Others argued that unknown migration events after Columbus could have introduced the lineage earlier than documented. So researchers expanded comparative databases. As global genetic datasets grew, the match to indigenous North American populations remained the strongest alignment. The haplogroup was not European. It was not Asian in the broader continental sense. Its closest relatives were across the Atlantic. This is where archaeology re-enters the story. Lance Meadows was not discovered through DNA. It was identified through structural remains, turf walls, ironworking debris, unmistakably Norse artifacts. Treering analysis later pinpointed activity there to 1021 CE. Using wood cut with metal tools indigenous communities in the region did not use at that time. That date aligns almost perfectly with the genetic convergence in Iceland. Coincidence is possible, but convergence across independent lines of evidence becomes harder to dismiss. Greenland adds another layer. The Norse established settlements in Greenland in the late 10th century under Eric the Red. Those settlements persisted for centuries before eventually disappearing. Excavations of Greenlandic Norse cemeteries have produced skeletal remains suitable for genetic analysis. As sequencing technologies improve, researchers are beginning to examine whether similar non-European lineages appear in Greenlandic remains. Preliminary findings suggest a more complex genetic landscape than previously assumed, though data remains limited. Preservation conditions in Arctic environments are variable, and sample sizes are small. But even a single confirmed case of indigenous American ancestry within Viking Age remains in Greenland would strengthen the case dramatically. Beyond individual lineages, there is also environmental DNA. Recent advances allow scientists to extract genetic material not just from bones, but from soil layers themselves. At Norse sites in Newfoundland, soil samples have yielded traces of plant species not native to that region, suggesting transport of materials across distances. If human DNA fragments could be recovered from these contexts and linked to indigenous populations, it would represent another piece of the puzzle. But caution is necessary. Ancient DNA research is notoriously vulnerable to misinterpretation. Early studies in the 1990s produced sensational claims, later retracted due to contamination. The field matured by learning from those mistakes. Today, replication across independent laboratories is standard. Statistical confidence thresholds are strict. Data is publicly archived for reanalysis. This is not a field driven by speculation. It is driven by verification. So what does the evidence currently allow us to say with confidence? First, Norse explorers reached North America around 1000 CE. This is archaeologically confirmed. Second, A mitochondrial lineage of indigenous North American origin appears in Icelandic populations, with genealogical tracing back to the Viking Age. Third, molecular damage patterns and phylogenetic comparisons support the authenticity and age of that lineage. What remains uncertain is scale. Was this a single individual transported to Iceland? Were there multiple voyages involving human exchange? Did some Norse remain in North America and integrate into indigenous communities? Genetics can identify lineage presence. It cannot reconstruct personal narratives. But even one confirmed case reshapes the narrative of contact, because it means the Atlantic crossing was not a one-way reconnaissance mission. It was a bridge traversed in both directions, and that bridge existed 5 centuries before Columbus. The resistance to this idea is not purely academic. Historical narratives anchor identity. The age of exploration, as traditionally framed, begins in the late 15th century. If sustained transatlantic contact occurred in the 11th century, even at small scale, then the discovery framework becomes less absolute. It becomes a matter of scale and consequence, not chronology. Columbus initiated large-scale colonization. That remains historically transformative. Here is where the story becomes uncomfortable. Because proving that Vikings reached North America is one thing. Proving that they changed something is another. Archaeology gives us structures. Genetics gives us lineage. But what both are hinting at is something far more volatile: sustained contact. And sustained contact leaves consequences. The sagas describe tension with the people they called Skraelings. Short encounters, trade that turned violent, settlements that failed because resistance was strong and numbers were small. That is the official interpretation. But sagas were written generations later. They were edited, framed, simplified. They serve cultural memory, not forensic documentation. And they do not describe everything. Think about what it would actually take for a Native American mitochondrial lineage to enter Icelandic society around the year 1000. Not a single encounter, not a raid and retreat. A successful return voyage across the Atlantic. That implies stability, knowledge of routes, confidence in navigation, repeatable logistics. You do not risk transporting a person across 1,000 miles of hostile ocean unless you believe you can survive the crossing, which suggests that by the early 11th century, the Atlantic was no longer an unknown barrier to Norse sailors. It was mapped in memory. Greenland had already been settled for decades. Eric the Red's colony functioned as a stepping stone, a launch point further west. From Greenland's western settlements, North America was not myth. It was reachable, seasonally, strategically. The site at Meadows was likely not chosen at random. It offered access to timber, something scarce in Greenland. It offered proximity to resources. It functioned as a base for exploration deeper into Vinland. And here is what most people overlook. Meadows is the only confirmed Norse site in North America. Not the only possible one. Archaeological absence does not equal historical absence. Coastal erosion, shifting shorelines, indigenous settlement reuse, and organic construction materials make detection difficult. What if there were more camps? Smaller ones. Temporary ones. Sites that left almost no trace beyond soil disturbance and decayed wood? We are searching for 11th century footprints on dynamic coastlines. It is possible that most evidence is already gone. But genetics does not erode as easily. If Norse explorers integrated with indigenous communities, even briefly, traces could survive in both directions. And that is where the investigation becomes even more delicate. Because if a Native American lineage entered Iceland, the reverse question follows naturally. Did Norse genetic markers enter indigenous populations in North America? So far, evidence is limited. Indigenous genetic studies are complex, politically sensitive, and ethically constrained. Many communities control access to ancestral remains and genetic data, understandably cautious of misuse. But preliminary analyses of certain northeastern populations have not revealed clear Norse signatures from the 11th century. That absence does not disprove contact. Small-scale exchange can leave asymmetric traces. If only a few individuals traveled eastward to Iceland, and none remained westward long enough to reproduce, the genetic signal would be uneven. The direction of travel matters. and the sagas hint that Norse settlements in Vinland were short-lived due to conflict. If Norse presence was temporary and numerically small, their genetic footprint in North America may have been diluted or erased over centuries. But even a single confirmed indigenous lineage in Viking Age Iceland shifts the equation, because it demonstrates return, and return changes the psychology of exploration. Columbus's voyage is often portrayed as an act of radical uncertainty. Sail west into nothing and hope to survive. The Vikings did something different. They extended outward step by step. Norway to Iceland, Iceland to Greenland, Greenland to North America. Incremental expansion. Each new landmass reduced the risk of the next. By the time they reached North America, they were not leaping into the unknown. They were following a chain. This makes their achievement less dramatic in popular imagination, but perhaps more impressive in technical terms. Their ships, clinker-built longships and Nar cargo vessels, were engineered for open ocean. Flexible hulls, shallow draft, capable of beach landings and deep water crossings. They did not need compasses. They needed experience. And experience is cumulative. Now consider what it means culturally. If Vikings brought a Native American woman to Iceland, she did not exist in isolation. She entered a community structured by kinship, law assemblies, oral tradition. Did she speak Old Norse? Did she retain elements of her own culture? Was she a captive, a traitor, a voluntary migrant? We do not know. But she survived long enough to have descendants, which means she was not erased. Her lineage endured through centuries of plague, famine, volcanic eruption, and political change in Iceland. That is not a footnote. That is integration. And integration implies human complexity beyond the saga shorthand of conflict with Skraelings. It suggests moments of negotiation, moments of curiosity, moments where fear did not win. History prefers clear categories: conqueror and conquered, discoverer and discovered. But the Viking Atlantic story may resist that simplicity. And this is where resistance intensifies. Because if transatlantic contact occurred in the 11th century, it reframes how we think about pre-Columbian global isolation. It does not mean the Americas were widely known to Europe. It does not mean maps circulated or trade networks flourished across the ocean. But it does mean that the Atlantic had already been crossed, survived, and reversed. The psychological barrier was broken long before 1492, and barriers once broken are never the same again. As sequencing expands and more Viking Age remains are analyzed, researchers are now asking sharper questions. Can isotopic analysis of skeletal remains reveal individuals who grew up in North America but were buried in Iceland or Greenland? Can trace element signatures in teeth confirm childhood geography different from burial location? Can environmental DNA in Norse sites in Newfoundland detect human signatures distinct from Scandinavian profiles? The tools are improving, and every improvement increases resolution. What once looked like a myth is now a dataset. What once sounded like nationalist exaggeration is now a molecular anomaly demanding explanation. But perhaps the most unsettling realization is this. History did not forget the Vikings reached America. It simply minimized it, because their contact did not explode into empire. It flickered, and flickers are easy to overlook, until science turns up the brightness. If future evidence confirms additional indigenous lineages in Viking Age Scandinavia, or Norse markers in early North American remains, the story will move from possibility to inevitability. And at that point, the question will no longer be whether Vikings reached America first. It will be how much of the Atlantic story we have misunderstood. Because sometimes history does not change through new discoveries. It changes because we finally learn how to listen to what was always there. Encoded. Waiting. In the blood. There's a moment in every historical shift when the evidence becomes impossible to ignore. We are approaching that moment now. For decades, the Viking presence in North America was treated as a fascinating anomaly. A confirmed landing, yes, but contained. Safe. Neatly boxed into a single archaeological site in Newfoundland. A bold voyage that ended quietly. But ancient DNA does not behave quietly. It expands the story beyond structures and sagas. It forces us to imagine people moving across the Atlantic, not as abstractions, but as individuals with names we will never know. And once you accept that even one indigenous North American lineage entered Viking Age Iceland, the entire Atlantic narrative destabilizes, because that implies continuity, not just arrival, not just survival, but return. Think about what return actually means in the year 1000. It means a ship left North America, navigated back to Greenland, then onward to Iceland or Norway, carrying passengers and cargo. It means those aboard expected land to reappear beyond the horizon. That expectation changes everything. Exploration without return is accident. Exploration with return is knowledge. And knowledge spreads. Even if no formal maps were drawn, sailors talk. Routes become memory. Distances become estimated. Seasonal winds become predictable patterns. The Atlantic becomes measurable. By the time Columbus sailed west in 1492, he was entering waters already crossed centuries earlier, not widely known, not systematized into empire, but crossed nonetheless. And here lies the difference that history has blurred. Columbus triggered transformation because his voyage connected continents in sustained, organized, imperial ways. The Viking crossings, by contrast, were small-scale and fragile. But small does not mean irrelevant. If Norse explorers established even temporary seasonal circuits between Greenland and North America, then for several decades, possibly longer, the Atlantic functioned as a corridor, not a wall. That corridor may have been narrow, but it existed. So why did it disappear? Greenland's Norse settlements eventually collapsed in the 15th century. Climate shifts during the Little Ice Age made farming harder. Trade with Europe declined. Isolation deepened. By the time Columbus sailed, Greenland's Norse communities were already fading or gone. If the stepping stone vanishes, the route collapses with it. Contact is forgotten not because it never happened, but because the infrastructure that sustained it failed. And without sustained contact, history forgets quickly. But DNA does not forget. The mitochondrial lineage in Iceland does not care that Greenland collapsed. It does not care that sagas were rewritten. It does not care that Columbus became the symbolic starting point of transatlantic history. It simply persists. And that persistence forces a reframing. This is not about dethroning Columbus as a figure of global consequence. His voyages undeniably reshaped the world in ways Viking expeditions never did. This is about chronology, because first carries psychological power. The idea that Europe's encounter with the Americas began in 1492 is deeply embedded in education and identity. Remove that singular starting point, and history becomes layered instead of linear. Layered history is harder to teach, harder to summarize, harder to mythologize, but closer to truth. The Viking story, reinforced by ancient DNA, suggests that transatlantic contact was not a sudden rupture of isolation in the late 15th century. It was a gradual testing of boundaries that began centuries earlier. And here is where the next frontier lies. Scientists are now exploring isotope analysis alongside genetics. Teeth, in particular, preserve chemical signatures from childhood environments. Ratios of oxygen isotopes reflect local water sources. Strontium isotopes reflect regional geology. If a skeleton buried in Iceland shows strontium ratios matching northeastern North America, that would be powerful corroboration of transatlantic movement. Preliminary work in Viking cemeteries has already revealed individuals who grew up outside Scandinavia, many from the British Isles, consistent with known Viking activity there. The methodology works. It is only a matter of time before similar analyses are applied systematically to remains from Greenland and Iceland dating precisely to the period of Vinland exploration. If even one individual shows childhood isotopic signatures consistent with North America, the debate will intensify dramatically, because then the story moves from genetic lineage inference to geochemical confirmation, and each additional layer reduces uncertainty. There is also another angle rarely discussed. What if the Vikings were not the only ones capable of crossing? Indigenous North American seafaring traditions were sophisticated along certain coasts. It is possible, though currently speculative, that exchange may have involved movement in both directions initiated by indigenous groups themselves. We do not yet have evidence for that, but absence of evidence in this context often reflects absence of excavation. The Atlantic world of the 11th century was more dynamic than we assume. Storm patterns shift. Ice recedes seasonally. Driftwood from North America routinely washes up on Greenlandic shores even today. Knowledge of Western landmasses may have circulated longer than preserved texts suggest. And when you place the genetic data into that broader environmental and navigational context, the picture sharpens. This was not a single heroic leap. It was a frontier. A frontier explored, tested, and briefly inhabited, then abandoned. History often privileges the event that changes everything. But sometimes the more revealing story is the event that almost did. The Viking presence in North America did not reshape continents. It flickered and faded. Yet that flicker carried across an ocean and left biological evidence durable enough to reach us now. Which raises a final question. If ancient DNA has already rewritten the margins of the Atlantic story, what else is waiting in the genomes of forgotten graves? Every technological leap in sequencing increases resolution. We are entering an era where entire migration networks can be reconstructed from fragments of bone no larger than a coin. What feels radical today may feel inevitable tomorrow. And when the dust settles, the narrative may look something like this. In the late 10th and early 11th centuries, Norse explorers expanded westward in incremental stages. They established Greenland settlements. They reached North America. They interacted with indigenous communities. At least one individual crossed the Atlantic eastward and integrated into Icelandic society. Her lineage endured. 5 centuries later, Columbus sailed west and triggered sustained colonization that permanently linked continents. Both statements can be true, but only one has dominated collective memory. Ancient DNA is not erasing Columbus. It is restoring the Vikings to the timeline, not as legend, not as rumor, but as biological fact. And once biology enters the record, history must adjust. The Atlantic was crossed before 1492. Not accidentally, not mythically, but deliberately. The longships came first, and their wake is still visible, not on the surface of the ocean, but in the code written inside us.